Let's start out with our learning objectives. We want to be able to define human osteology in this module. We've looked at that. We do have those definitions, so that will reappear. We're going to talk a little bit, and we want to be able to recall our evolutionary and biocultural connections that we see in the skeletal system. We want to be able to refer to and define our anatomical terms of reference. So for example, those terms that we're going to hear like ventral and dorsal, superior, anterior, etc. We're going to recall some of our bone growth patterns and related terms, things like epiphyseal ends. We're going to know the names of our bones in the body and the various types, for example, long bones versus short bones. We're going to be able to identify major landmarks that are used to estimate identity. We're going to be able to side bones and recall how our bones articulate or how they fit together. And we're going to be able to apply this knowledge in our final assignment. When we're looking at bones or any remains that we find, there's a number of initial steps that we're going to go through, and we can start off, is it blank? Question mark. Well, the first thing is it bone? In some situations, we do find material that looks a heck of a lot like bone, and it may deceive individuals who aren't versed in human osteology. So that's going to be number one. We're going to want to take a look at that, say, is it bone? Secondly, is it human? So looking on the right-hand side of the screen, we have two bones. Are these human bones? The first ones that we see on the left side. These are not human bones. These belong to a bear. So there are a number of homologies. And we've talked about that. We've defined that in our first module. Between animals, those evolutionary connections, and our bones do look similar in some situations. So a zooarchaeologist, a bioarchaeologist, a forensic anthropologist, a human osteologist will be able to distinguish between, for example, on the left-hand side, bones that belong to a bear versus the bones that belong to a human. So here we have a human tibia. We want to look at those remains and determine whether or not they are modern or archaeological. In other words, do they relate to forensic anthropology? Are they recent? Or are they archaeological in their context? Do they date back prior to our modern forensic inquiry here? And we're going to generally look at the type of bone that we have, the deposition or how far it is in the ground, and we're going to make that assessment. We're also interested in what's referred to as MNI, the minimum number of individuals. So we count bones, and if we find two, for example, two right tibia, well, our minimum number of individuals would be 2 people. And we're also going to take a quick look at those remains and see if there's any evidence of trauma. And that's going to be particularly important if we're looking at a forensic case. And it's also going to be equally important depending upon the type of research that we're doing in archaeology or bioarchaeology. Okay, so osteology, it's a subdiscipline that can fit into anthropology, zooarchaeology, paleontology, and if we're just looking at this word osteology, it refers to the study of bones. Human osteology, in contrast, we're not interested in zooarchaeological remains, and I've pointed out that zoo, it's derived from a Greek word, zoon, which means animal. And while we are animals, here they are referring to non-human animals. Paleontology, that would be an area that's interested in non-human remains that go back very far in the history of the Earth, for example, dinosaurs. With respect to human osteology, then, it's the study of bones, skeletal elements, bone pathology or disease, the function of bones and how they change across populations, or what people previously referred to as race or ancestry. So we do have different functions to bones, just in the same way that we have different tones of skin color. We know that that has an evolutionary or provides an evolutionary advantage to certain populations depending upon the climate that they evolved in. So bones, we're going to see that same type of variation. Now, if I asked you, and I might on a test, what field does human osteology fit into? We need to say, well, it fits into that biological or physical. Remember, they're the same thing, biological or physical anthropology. And who uses human osteology? Forensic anthropologists. And we have bioarchaeologists. And within this area, we're also going to have, generally speaking, our evolutionary anthropologists as well. So a strong understanding of human skeletal anatomy is important to all of these subfields within anthropology. When we look at skeletal remains, we always place them in anatomical position. So the individual, the drawing that we see on the right-hand side of the screen, this individual is in anatomical position. We're looking at the front of their body. Their hands are showing us the palm. And think of Leonardo da Vinci's Vitruvian Man. That is also an anatomical position. From anatomical position, we use a number of terms of reference. So it allows us to talk about certain sides of the bone. We don't say the left side, the right side of the bone, or the front or the back. That would be lay terms for that. We use words instead. For the front, we'd say anterior. For the back, we'd say posterior or dorsal. So our anatomical terms of reference then, they include anterior or ventral. They're the same, they mean the same thing. It means the front. I'll see if I can squeeze that in here. So the front in anatomical position. In contrast, posterior or dorsal in anatomical position means the back side of the bone. So if I drew, for example, really quickly here, I'm going to try and draw a scapula or a shoulder blade from an anterior perspective, or the front. So this would situate right here in our body. We have a flat surface. If we looked at the dorsal side of it, and I'm going to try and flip it around, see how good my skills are. So we have something like that. We have a spine that runs down the back. That would be the dorsal side, the back side. And the area that articulates with the humerus, right here on the anterior side. And if we were looking from the dorsal side, we'd be looking at it on the opposite side. This is important because it allows us to side our bones and know exactly what we're looking at, and especially if we're dealing with fragmentary materials. Proximal and distal. So a couple of other terms. Proximal means closer to the head, whereas distal means further away. So if we're looking at, say, I'm going to try and draw a humerus in here. So our humerus would articulate as such. And again, remember, we're looking at the dorsal view here. We're going to say that the proximal end of the humerus, or the humeri, is closer to the head, Whereas the distal, so we can put a proximal up here, the distal refers to the portion of the bone that's further away from the head. Lateral and medial. So if we're looking at this center line, this red line that's moving through the body, an anatomical position, medial means closer to the center line, whereas lateral means further away. So we see it at the bottom, we see lateral, it's further away from the center. And if we're looking at, and again, we need to orient ourselves here in some situations, so let's go back to our posterior drawing of the scapula and the humerus. We're looking at the backside or the posterior, the dorsal view. Medial, if we drew that red line in there again, it would be right about here. Okay, and because this bone belongs to the right side of the body, we're going to say that here is the medial, and over on this side would be the lateral portion of the bone. So the head of the humerus here is medial. In contrast, if we move over to a tubercle on the side, the greater tubercle, that is lateral. Superior and inferior. Superior means above. Write that in. And inferior means below. Okay. One of the things I want to point out here is we need to use these terms in our assignment for full marks. We will be looking for that. So if you're talking about a portion of a bone where you may see, I'm going to just use green to highlight an area. Say we have a lesion here, a hole in the bone. That's something that you want to note. Maybe it's a bullet hole. Maybe it's a result of bone remodeling, an osteolytic lesion that's been eating away at that bone. We want to be able to say where it is on the bone. So I'm going to just color that in here, and I'm going to use green here. So a few of the things that we'd want to say here, it's on the humerus, and we're seeing it located medially. And it's distal. Okay, so it's medial, it's distal, and it's located on which humerus? So again, we need to think that this is the back. So if we kind of swung it around, it's going to be located right here. So it is a humerus. It's a right humerus. The location of the particular feature that we're looking at, it's medial, and on the distal end of that humerus. And we perhaps can point out one more thing here. It's also dorsal. So this allows us to say it's on the back side of that humerus. It's located towards the end of that humerus. So the back side of that humerus. I'm just going to highlight here. So dorsal, it's on the back side of that humerus. Distal, it's towards the end of that humerus. And medial, it's towards the center of the body. And again, that's just a common way to look at bones and know exactly what we're talking about. So make sure that you use these in your assignment. Okay, so there are a number of different types of bone. I'm just going to sketch in here something, and hopefully I don't go over top of my drawing. Let's go again, consider that humerus. And let's look at an anterior view of the humerus. In other words, we're looking at the front of that humerus, and because we see the head of the humerus located medially, okay, so it's medial in this particular situation, and it's the front, it's pointing inwards to that line, we know that we're looking at a right humerus. Just a little bit of review. And the humerus, it's a long bone. In contrast to the humerus, we find some bones, for example, I'm going to try to draw it by scale here. So if we go into our finger bones, we have what's referred to as our metacarpals. If a humerus is a long bone, what, pray tell, is a metacarpal? You're probably saying, well, it's a short bone. No. It's also a long bone. So a long bone is defined by the length versus the width of the bone. So here our length, I'm going to try to draw this in, squeeze it in, doing a lot of squeezing, much more difficult than on a whiteboard. So our length is greater than the width, and that's what we use to define these particular bones. So we have really 5 types of bones here. We're going to be interested in really 4 of them: our long bone, our short bone, a flat bone, and an irregular bone. We also have sesamoid bones as well. So I want you to be familiar with be able to define at least the first 4: long, short, flat, irregular. So a few examples of these. Let's go to long bone because we're working with that already. So I'm going to highlight that here with an asterisk. Our long bone, femur, great example of a long bone. We're seeing that width isn't as great as the length. That's a long bone. In contrast to that long bone, we have a short bone, and a short bone is defined again by that relationship. So let's go over here to the, get a good view here, the intermediate cuneiform, where the width and the length are generally the same. Not much of a difference. So that's going to be our short bone. The width and length are mostly equal. Our flat bone, right up to the top, this is a bone really that just is that. So we're looking at the sternum and the manubrium. These bones are flat, so if we looked at a profile of this bone, it has this type of structure. We find a lot of flat bones in our skull. As for irregular bones, these are bones that have irregular shape, and vertebrae, or the vertebra. One particular example of the vertebrae, the vertebra, it has an irregular shape, and we use that term. As for our sesamoid bone, we have our patella is a good example of that bone. The human body also has 2 basic types of skeletons that are distinguished within it. So we have the axial skeleton and the appendicular skeleton. The axial skeleton, here I'm gonna continue to use red, I guess. That's all right. The axial skeleton, it includes the skull. It includes the spinal column, or the vertebrae. We have our ribs are included in that. We have, can't really see it very well here, but the sternum, that's part of it. And down to the sacrum. So this is kind of the central or the axle of the skeleton. In contrast, the appendicular skeleton, it's going to include everything else, including the clavicle, the scapula, our humerus, our ulna, our radius. I'll go through all of these terms, don't worry. The innominate bone, we have our femur, we have the patella, the tibia, the fibula, and the bones in our feet and our hands. So our carpals in our hands, our metacarpals, and the phalanges. And in our feet, we have the tarsals, the metatarsals, and the phalanges as well. Okay, so how many bones do humans have? The immediate answer might be, well, we have 206 bones. Well, depending upon the question, And remember, one of the key features that I pointed out among humankind or any animal or plant is variation. So we need to be able to recognize that for a test. We want to be able to say, generally speaking, an adult, generally speaking, has 206 bones. But because there is variation, sometimes we do see extra bones. And quite often these come in the form of wormian bones. When we're born, we have over 300 bones. And over time, these bones, they solidify or they fuse, producing that 206 on average bones that we find in a human adult. Okay, so looking at the bone plates and development, and here we're specifically looking at subadults. Because by the time we get to adulthood, our bones are fused. Answering the question, how many bones at birth? Plus 300. And why? Well, really, this is an evolutionary thing. If our bones weren't fused, we wouldn't be able to progress through the birth canal. And because we are bipeds, that reoriented the pelvic girdle. It made it smaller in comparison to other animals. So if we didn't have plus 300 bones and all of our bones were solidified, birth wouldn't be possible. So our bones are not fully fused at birth. And we want to be able to break some of these bones down here, at least our long bones. We want to be able to take a look at them and say that they have three main components, because this is going to be important when we do take a look at some bones. Some of them may belong to subadults, and we want to be able to distinguish a bone that belongs to an adult from a subadult. Here we're looking at an adult humerus, and I'll show you a subadult bone shortly. We're looking at the adult bone that's fully fused. But we can take this and break it up into three sections, and two of them are repeated. So the proximal end of this humerus, I'm going to sketch this in here for you, the proximal end here, so we're highlighting it in the green section. This is the epiphysis. Moving to the gray section, the metaphysis. It's a funny word, I'm sorry, it's hard to pronounce. And the brown section, we're looking at the diaphysis. And then again, the distal portion of this bone, the metaphysis, and then the epiphysis. Unfused areas, they create these plates that I've referred to as epiphyseal plates or growth plates. And I'm going to show you an image of that. Now, that's something that we can see when we're looking at remains. It's very noticeable. And even when we look at a skull, for example, hopefully I have the room here and something doesn't pop up. If we look at our skull, draw it in quickly. We have a number of suture lines that you're probably familiar with. Everybody draws this type of stuff in, but they kind of put them everywhere. We have a bone here that we call our frontal bone, and then off to the side we have something called our sphenoid, and then we have our temporal bone, and back here we have our occipital bone. So we have these sutures, and they will become solid over time. And even if we looked at, say, an anterior view or ventral view of a skull, one of the things that a lot of people don't know, and again, we can draw some of these in. We're not going to see a whole lot because we have our frontal bone. We do have a coronal suture up at the top. And there's our frontal bone, we can't see the sphenoids and we can't see the temporal bones here. We do have a suture right there, for example, because our maxilla, it is two pieces at birth, and if I added in a mandible, we also have a suture there as well, and that fuses as we age as well. Okay, so here we have an X-ray of a couple of long bones. We see 3 long bones. Can you identify them? I'm going to write them in at the top. See if you can write it down. So if you thought that the top one was a femur, you're correct. Right there, that's our femur. There's our tibia and the fibula right there. And let's go ahead and write in the sections of our bones for review. There we go. Okay, so those are parts of the bones, and we can see a good example of the epiphyseal plate. I'm going to go in with some white here, and we see an epiphyseal plate here that exists between the epiphysis and the metaphysis. Get rid of those. So these growth plates, they are visible not only through X-ray, but when people find remains, archaeological remains or forensic remains, this is one of the things that people are looking for. So our individual bones, they fuse within approximate age spans. So in other words, based upon the type of bone that we're looking at, and refer to your WordPress here, readings on this, we do have some dates that I want you to read up on. Based upon the bone that we're looking at, there are particular growth patterns that we see, and it allows individuals that are doing forensic anthropology or bioarchaeology to estimate the age at death. Here's a question. What do our readings say about the clavicle? This would be a good question for the test to see if you're reading along. I won't tell you. I'm going to leave that out there. I'm going to let you research that. Okay, so our known fusion times, as I pointed out, they're used to estimate AAD, age at death. So with respect to our WordPress, I'm not going to tell you about the clavicle. You're going to research that, but here I will give you a couple examples of the process that we see. So in subadults, for example, 16 months of age, and if you're wondering what this is, we're looking at the sternum, and because there's only one sternum, we don't need to say it's right or left, and we're looking at a ventral or anterior view. So at 16 months of age, we see that the bone that will eventually become one bone that we refer to as a sternum, we find it in 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10 parts. So this is one of the reasons why we can get up to that 300 plus bones at birth. By the time we get to just around or just under 4 years of age, less than 4 years of age, we see that these sternebrae, that's what they're referred to as, the sternebrae, that they've fused, giving us, instead of 1, 2, 3, 4, 5, 6, 7, 8 sternebrae that we would find in this section here, they've fused into 4 separate units, and what eventually becomes a xiphoid process that stays. So now we're down to 6 main components just under 4 years of age. And we see further fusion, leaving us again with, let's look right at this point, vertebrae 3, sternebrae 3 and 4, they become fused between 5 and 10 years of age, leaving 1 and 2 still unfused by that age. So when an individual is discovered and that sternobray has fused, that allows the researchers to say that we're looking at approximately 5 to 10 years of age. And that's an approximation, but it allows the researchers to continue to figure out what's going on. So moving a little bit further on, we're going to see by 11 to age 16 or plus 16 that our sternobray 2, 3, and 4, so 3, 4, they've already fused, but number 2 here is fusing, so now we have fusion between 2, 3, and 4, and our sternobray 1, it's still unfused, and it's not fused well in 2 points with the manubrium on the top. Just put an M there, and with sternobray 2, 3, 4. By the time we get to 16 to 20 years of age, that we now see that all of the sternobray, they have fused. So sternobray 1, 2, 3, and 4, they're fully fused by 16 to 20. We don't see fusion with the xiphoid process as of yet, and we don't see fusion with the manubrium. 40 plus years of age, now we see that xiphoid process. It has become fused, and in a lot of cases we still don't have full fusion with the manubrium. After that, we will start to see that manubrium fuse with now what we refer to as the body. So the anatomy here, and again, so manubrium, and we got a manubrium, we've got the body, and down on the bottom, I'm not going to be able to squeeze it in, so I'm going over to the left-hand side, the xiphoid process. I'm just going to put an X there, the xiphoid. I'm not going to go through this in depth, but again, even when we look at that, the innominate bone, or what eventually creates the pelvic, I shouldn't say eventually, it does create the pelvic girdle. Sometimes it's referred to as the os coxa. We use innominate bone. We see that at birth we have a number of clear parts to the innominate bone. For example, the ilium, so this is kind of our hip bone up top here. We have the pubis, so this is towards the front of our body, the ischium, that gets sore when we ride a bike. And we can see those areas that will eventually fuse. I'm going to try and use I'll see if I can go with yellow. See all of these areas here, they will eventually fuse over time. And one final example. So if we're looking at the femur, we see a number of different components throughout the life course, from unborn, newborn, juvenile, subadult, adult, to an elderly individual, where we see full fusion. So even a subadult, so we're looking at a teen here, where we're not seeing, and this is really why a lot of kids don't break their bones in the places that adults eventually do, because they are in fact malleable. So we see that those areas are not fully fused during while we're teens, and as we become adults, we're seeing full closure, almost full closure, depending upon the age in those particular epiphyseal plates. Okay, moving on to the skull. We want to be able to break the skull into actually three separate components here, because we don't include the mandible. So here, the colored areas, we're looking at what's referred to as the neural cranium. The neural cranium, the major bones that we need to be aware of, and I'm going to go through these in greater depth on a sheet for you. We'll write those out. The frontal bone, the parietal bones, temporal bones, occipital, sphenoid, and ethmoid bones. So here I'm just going to annotate next to these so we know how many they are. There is one frontal, we have two parietals, we have two temporals, we have one occipital, we have one sphenoid, and we have two ethmoid bones. The function of our skull is protection. It protects our brain. And interestingly, we do see some dimorphism or some differences between males and females when we look at the wall of the bone. I'm just going to jot that in here. So the anthropological symbol for male is triangle. They have thicker kind of funny. Males, no surprise, have thicker skull bones. And again, that's connected to our evolutionary past. And you know, divisions of labor, to some extent, they always have existed by necessity. So, for example, males, historically, they would have played a different role at various points of time with respect to hunting, especially at times when females, for example, and by nature, they gave birth. And because we're mammals, breastfeeding, so there would have been a little bit of a difference in the type of resource gathering that we would have engaged in historically between males and females. And that's not to say that women didn't engage in hunting. We know a whole lot about the past, and women did engage in a lot of hunting and gathering as well. But at various times, obviously, there's some differences there. Some of the landmarks that we're going to need to be familiar with, and there are a number of landmarks, we'll go over those in great detail. The landmarks that we have are specific regions on the bone. So, for example, I'm going to try and grab it right now. So on the temporal bone, the one that's a copper type of color, it's a little bit fast, and there it is right there, and now it's gone. And there it is right there, and we'll see that in greater detail. The mastoid process, for example, provides us with insight into whether or not an individual is male or female. It's used to estimate because males tend to have larger mastoid process, and here I'm using the word tend because there's always a scale that we estimate on. There's always variation, but males tend to have a larger mastoid process. And when we use the number of these features or these landmarks in comparison, and this is a very good percentage to know for a test, we can achieve 80 to 90% accuracy when estimating biological sex. Okay, so let's move on to the next part of the cranium. The visceral cranium, the section that we see highlighted here, colored in, that refers to all of those other components that we do not find in the neural cranium. So the major bones that we're interested in the visceral cranium include the nasal bones, and I'll annotate that as well. So we have two nasal bones, two lacrimal bones, two zygomatic bones. At birth, two maxilla, which eventually fuse, and giving us one maxilla, and our palatine bone, they fuse as well, eventually leading to one palatine bone. The mandible, as I've pointed out, when we look at the skull, I'm going to write that in up top because I've said it. The skull, I said, had three components. Okay, we have three components. With respect to the skull, we have the neural cranium and all the bones that we find in that. We have the visceral cranium and the mandible. So here, when we're looking at the cranium, where can I get that in? I want to say mandible is part of the skull, but not the cranium. So if we use this word cranium, we're just referring to everything excluding the mandible. The function of our viscerocranium, it's structural support. It provides our face. Ultimately, we do see some evolutionary differences there, pardon me, over time. For example, evolutionary comments, I'm going to point out the mandible. So the mandible is smaller today. At one point of time, it was, in fact, much larger than it was. We don't need the type of mandible that we had historically for mastication or chewing. So over time, a smaller jaw or a smaller mandible was selected for. And this change in our visceral cranium, the smallness of that, it also allowed for the development of a larger brain within the species, the evolution of the species. And again, we're going to find a number of landmarks that we're going to be interested in. And as we've pointed out, some of these areas combined, they can be used to achieve, we're missing achieve there, that's okay. Right there, to achieve 80 to 90% accuracy. Now that's pretty good. And again, keyword here, combined. So if you're putting together your assignment, you don't want to just look at the mastoid process and say this individual has a mastoid process that's representative of what we would see in a male, therefore it's a male with 90 to 80% accuracy. No, that's only one single part. We need a number of parts, a number of landmarks that we want to assess, and we're looking for dimorphism, or physical differences. Dimorphism, physical differences in structure and size, and that allows us to eventually make that estimation about sex in some situations. Other things that we can use it for would be also ancestry. And as we get further along in our lectures on human osteology, I'll point those out. Okay, so we are looking at a side profile or a lateral view of the skull. And we'll go through this and identify all the bones that we can see. Okay, so let's start off with this bone right here. That's our parietal bone. We have two of those. Right here we have our frontal bone. We have one frontal bone. We can see that. Our temporal bone. I'm just going to, when I can, I'll kind of highlight the sutures here. So we have a suture right in here. And as such, we see a suture right there. And we see a suture right there. There's another suture there. Okay, and another little suture in here. Continue that. Okay, that gives us a good idea. So we have our parietal as pointed out, our frontal. This is our temporal. And we have two temporal bones. Back here, the occipital. We have one occipital bone. Okay, right here. We have the zygomatic bone. We have two zygomatic bones. Our mandible, we have one. We can see our nasal. We have two nasal bones. Right here, we can see the better view of the lacrimal bone. Two of those. We can't see the ethmoid bone from this perspective. And we see the maxilla, and we have one of those. I haven't drawn in the sphenoid. I'll put in one more suture here we can see as such. And there's our sphenoid bone as such. Okay. From this view, we do have a view of the teeth. We can go through that 2, 1, 2, 1, 2, 3. So this individual, they do have that wisdom tooth there, and they have that normal hominid dental formula. 2, 1, 2, 3. Okay, let's move on. We can see one more bone a little bit better from a different view. Okay, so here we are looking at the skull, and we're calling it a skull because we do see the, yes, you're right, mandible. And our view is anterior or ventral, which means it's a front view in anatomical position. One of the things I'll sketch in here really quickly is sometimes we also see a suture here. That's referred to as the metopic suture. Again, just going back to the idea that our bones, and especially in our skull, they're not entirely fused at birth. So the frontal bone, it is in two pieces, and we also see a persisting suture in the maxilla as well. Okay, so here I'm going to go through all the bones that are visible. So starting off with this bone that's at the front. Perfect name for it. It's called the frontal bone, and we have one frontal bone when it's sutured. So when I'm going through these bones, I'm going to use the adult sutured number for how many bones we have. At the side of our skull, so we see a suture right around here. I'm just going to draw in a couple of points, and in between them you can see that suture line. This is called the parietal. And we have two parietal bones. Moving down a little bit further, right around our ear, you can feel that bone there. That's called the temporal. The temporal bone, we have two of those. Moving inwards or to ventrally, we have a bone that's called the sphenoid, and that progresses, that makes a part of our eye orbits, and it is one bone, so it goes from one orbit to the other bone, and it also represents, if you feel your skull right at your temple, that's where your sphenoid is. So we have one sphenoid. Moving a little bit more inferior from our sphenoid, so down, we have our nasal bones. We have two of those. We have our zygomatic bone. We have two of those. And then we have our maxilla. And we have our mandible. And one each. Okay. So these are, these are the bones that we can see from an anterior position. Oh, you know what? We can't see it, but we normally would be able to see it right where your tear ducts are. So I'm going to go to the other side. Right where your tear ducts are, we have a bone that's called the lacrimal, which means to tear, lactate, lac. So that's our tearing area. And then a little bit further back, we have the ethmoid. We have two of those and two of those. As for our teeth or our dentition, normally what we do is we divide our teeth into 4 quadrants, And you have your maxillary arcade. Just going to write arch, maxillary arch, and the mandibular arch, dental arcade. And we find in each quadrant the same teeth. So I am going to try to do this in a logical manner. I'm going to continue to use red, and I'm just going to highlight these as I go along. So our two teeth in the front in this quadrant, or any quadrant, we refer to as our incisors. So in any given quadrant, we find two incisors, and our total complement would be 1, 2, 3, 4, 5, 6, 7. You've got it, 8. So that's our total. The total number of incisors that we have is 8, but in each quadrant 2. And I'm just going to put that over here. So 2 incisors. Moving on a little bit further, we have a tooth that's referred to as a canine. And we have 1 canine in any given quadrant, giving us a total of 4. Let's go over here, 1. And then we have 1, 2, we can't see them really well. We have 2 premolars in any given quadrant, giving us a total of 8 premolars. And now we're having great difficulty seeing it. We're going to go back to our molars. And depending upon, I guess, your own evolution and the genes you have anywhere from 2 to 3 premolars. So the wisdom tooth. Some people, the wisdom tooth is congenitally absent. So we're going to put 3 here. And if people, well, they don't, not all people have them pulled, but our total would be 12. So really this lends us to a dental formula where we have 2 incisors, 1 canine, 2 premolars, and 3 molars. And this is also a dental formula that we find, for example, among gorillas. We find the same dental formula among chimpanzees or any other hominoid species that's related to us. Some individuals, for example, me, my dental formula is a little bit different from the average. It's 2, 1, 2, 2. I have a congenitally absent molar. So the wisdom tooth, they're not there. And that also is an evolutionary feature that ultimately, I'm going to write it down here, might come up later on, lack of wisdom equals more space for brain. So this is something that has evolved more recently as the human brain continued to increase in size. So let's go to a different profile here. And again, we have our skull, and we want to make sure that we label this. We're looking at a posterior or dorsal view. And the bones that we can identify here, we see a parietal. And I'm just going to identify the other parietal because we can see it well. And we have our occipital. And we know that we have 2 parietal bones. We have 1 occipital bone. We have our mandible. We can see well from here, from this perspective. We can see, okay, so here we can see part of the temporal. And as I pointed out, we have 2 temporal bones. And what we're seeing here in particular, I'm just going to highlight this Okay, so this landmark, I'm going to use a different color here. This is one of the landmarks or the processes that we see on the temporal bone. It's called the mastoid. And that's going to be important when it comes to estimating biological sex and estimating ancestry. Other things that we can see, well, we can see a little bit of the palatine bone right up in here. We can see our teeth. Again, a little bit more difficult for us to do, but we're seeing that 2, 1, 2, 3 dental formula. And we should, because, well, this is the same individual. The other thing that we're seeing, and remember I had said, generally speaking, humans have how many bones? 206 bones. But as Charles Darwin had pointed out, variation is a predominant and necessary. Right? Charles Darwin said that variation is a necessary feature in biological life. This is how new features and new adaptations emerge. So mutations, and if those mutations provide an advantage, they'll be selected for. They will remain within the species because it allows them to reproduce at a greater rate than individuals that don't have that trait. In some instances, we just see we see variation that really doesn't serve any purpose, but it is perpetuated in individuals. Here, what we're seeing is something called a wormium. Those wormian bones. So when we said generally humans have 206 bones, we said sometimes we see individuals with wormian bones. And when we see one in particular in this area, and we're not really seeing a great example of it here, in some situations we see an individual, I'm just going to redraw the suture, say it looked like this, right there, like a triangular bone. That would be called the Inca bone. And the Inca bone is often used to help identify ancestry. We tend to find it in Asian individuals, Asiatic, and also First Nation. So our First Nation peoples also tend to exhibit this as well throughout the Americas, not just in North America. I'll just erase that there so we can see a better example. But here we have a number of wormian bones, these little bones here. And sometimes that does give us insight into ancestry as well. And especially when we see fairly complex suture lines. This is a fairly complex line. And sometimes that's used to help identify ancestry. Individuals that are non-European, for example, Asiatic, African, tend to have more complex suture lines. Other thing I want to point out while we're here as well, I'm going to take advantage of this. We're going to come back to it just so we see it now that we're done with all of the red. So remember the red. I'm just going to write that up here so we know the red. That's all of our bones. And the white we've used to identify a couple of landmarks. At least here, this is our key, just so we don't get confused. The other thing I wanted to point out was this area that we're seeing right here on the occipital bone. This is called the nuchal crest, sometimes referred to as the occipital protuberance. The occipital protuberance. That's an ugly word. We'll use nuchal crest. And this is also used to identify whether an individual is male or female. Okay, it can be very helpful. All right, so now that we're done with the skull, let's take a look at a few more other landmarks and anatomical um, areas. Okay, so landmarks. Our bones, we need to know those major bones. I wouldn't be surprised. You shouldn't be shocked. You should be aware that on the test you'll see a skull and it'll ask you to label a particular bone. Is it the frontal bone? Is it the temporal bone? Is it the occipital bone, for example, or the maxilla? Here, one of the things that we do need to be able to apply in our assignment and on the test as well is to be familiar with some of the landmarks that allow us to estimate sex, ancestry, and any other features that we might be interested in helping us achieve positive identity. And remember, one of the main ideas is we are reconstructing life right from the skeleton. And we can do that here. So a few things I want to point out. I'm going to take a look at very quickly here at this individual, and same individual. So here we see, I'm just going to write this in. It's always great for us to review. We have a lateral and a ventral perspective or vantage. And we're looking at a male. There's no question about that. From this, it leads to that, as I pointed out, 80 to 90% accuracy. I'm looking at a few areas, and I'll use yellow as well. So the first area that I'm taking a look at here is the supraorbital brow ridge. And that's a landmark. I'll write that in. Or a process that's used to help reconstruct life from the skeleton and achieve positive identity. So here we're seeing a fairly large supraorbital brow ridge, just beneath your eyebrows. Some people have large ones, some people have smaller ones. If we move a little bit further to our right, or we're starting to move to the dorsal, I'm going to use red. I've used that already. Sorry. So dorsal. We're still in a lateral view here, but we're moving dorsally to the back. Go back to yellow. What we're seeing in this area, and I pointed this out. Well, that's not a great place. I'm going to go over here. We see a mastoid process. So this is the mastoid process right in this area, just below the auditory meatus. So you can feel that if you go to your student famously commented, he said, the auditory meatus. Do you mean the ear hole? Yes, I mean the ear hole. So take your finger rested on your ear hole or the auditory meatus and just slide it down and dorsally a little bit to around your neck area. And you're going to feel a protuberance. You're going to feel a bone sticking out. That's part of your temporal bone. It's perfectly normal. And that is helped used to identify the sex of an individual. It exemplifies dimorphism. Okay, so moving to the dorsal area, to the occipital. Okay, so the mastoid, we're going to say, let me write this in. We find the supraorbital, we got that on the frontal bone, right? The mastoid, we're finding that on the temporal bone. And on the occipital, okay, so right at the back here, we find that nuchal crest. And here we do see it sticking out, right? And that's a muscle attachment. So that's a point for muscle attachment for the neck. And that's why we see some of the dimorphism. So if I drew in some of the muscles here that we have, depending upon how muscular an individual is, and this can increase in size over time depending upon an individual, how much exercising they engage in, how much weight they're lifting, what type of labor they're doing. So we can even use some of this stuff depending upon where it is on the body to determine whether or not an individual is right-handed or left-handed. Either way, this helps us say, well, one of the things that we know is one of the basic differences between males and females. Males tend to, and here I'm using that word tend, they tend to have more muscle. And historically, they've engaged in more of those laborious, not that the work of women is not laborious, but manual labor work. And that helps us identify whether or not somebody is male or female with a fairly good degree of accuracy. Moving to the ventral view, okay, on our mandible, we find something that's called a mental eminence. Like I said, that's on the mandible. And we see it right here, right, that area there. A chin, that's what it is. And finally, right here, okay, I'm going to try and squeeze this in, big word, superciliary arch, orbital arch. And that is located on the frontal. And what we tend to find with males is it's more robust. So that's one of the words. You'll be introduced to these words later on, but it's a robust feature. And when we tend to see robust features, that allows us to say that it's closer to the dimorphism that we see within males. In contrast, we use the word gracile if we're referring to soft features that are more consistent with the female sex. Okay, what we're going to do now, this was just an introduction. I'm going to go through all of these because, and I'm not going to teach you how to estimate sex. I'm not going to teach you how to estimate race at this point in the course. That will come a little bit later on. But I do need you to know those areas that you want to focus on when you're collecting data. Okay, let's move on. All right, I've provided you with these resources, so you should have them printed off. If you haven't done that, print them off and work along with me. Important to keep your notes with you and have well-organized notes for our test. So, here we're looking for a few landmarks, and the landmarks that we're interested in, we're going to find, and some of them are just by the name of the bone. So frontal bone, and I'm just going to write in bossing. Okay, so bossing on the frontal bone, obviously we find that on the frontal bone, and it helps us identify sex. And it doesn't help us with ancestry. The gallbella, we find that on the frontal bone as well. That helps us identify sex. Supraorbital brow ridge, that is also found on the frontal bone, and we're going to review all of this on the next slide, exactly where it's located. That helps us identify the sex of the individual as well. Orbital ridge, it is found on the frontal bone, sometimes referred to as the superciliary arch. Write that in there. And it also helps us with sex. The orbits. Okay, so the orbits, they're a composite of our frontal bone. I'm not going to write all of this in, but our frontal bone, we have our sphenoid, we have our lacrimal, we have the ethmoid, and it helps us identify ancestry. Okay, our mastoid process, that's found on the temporal, and that helps us identify sex and ancestry. So what I would suggest is you make sure that you're going through this. You're going to have your notes, and when you're collecting your data, you're going to make sure that you take pictures of these areas. And again, note, okay, I have the mastoid process. It's an area that I want to be able to analyze when I'm completing my assignment, and it might even, writing that in might be part of your draft work as well. The auditory meatus, we do find that on the temporal bone as well, and that helps us identify ancestry. Zygomatic extension. So this is on the zygomatic bone as it extends to the temporal bone. So we're just going to say zygo to temporal. So two bones involved there, and it helps us identify the sex of the individual. The nuchal crest, you're going to find that on the occipital, and it helps us identify sex. The inca or the wormium bones, we're going to find that on the occipital bone as well, and that's going to help us with ancestry. So when we begin to compile all of this information and we're taking it all into consideration at the same time, not just one of these features and saying, I see a large gallbella, so the individual must be male, that's not a logical conclusion. When we look at a number of these and we assess them, it allows us to place that individual within, as I pointed out, that 80 to that 90% accuracy. Okay, so we're back to our friend the skull here in two views, the same individual we were looking at originally, and maybe we can come to an assessment at some point of time just for fun. Like I said, we'll teach you a little bit more about that later on. So number one, we have the frontal bone, and as I pointed out, we're looking for bossing. So here we can say bossing and slope. That's the main feature that we're looking at on that frontal bone, or normally what we refer to as the forehead. So what are we seeing in this area? Embossing just refers to the bumps that we see. So here we see a little bump here, a little bump there. Okay. Moving down a little bit further, we have the gallbella. Okay, so from our table, this is corresponding to that table that we just wrote up. And depending upon whether or not it is robust or not, gracile versus being robust, that's going to help us determine whether or not we're looking at male or female. Number three, I'm going to go over to the right-hand side. This is our supraorbital brow ridge. And again, depending upon the size, whether it's robust or gracile, so the comparison is always important here, that we look at a known male, a known female, and make our determination based upon that. So the supraorbital brow ridge, if it's robust, then it's going to fit within a male assessment. Number four, I'll go back. No, I can write on this side. This is the supraciliary arch. And what we're doing is we're just kind of feeling inside, seeing how thick that aspect or that area of the frontal bone that's making up the orbit, how thick is it? Right, and again, we can see it over here, this area. Is it thick or is it fairly narrow? If it's thick, that's something that we find consistent with males. If it tends to be fairly small, and remember I'd pointed out some of those differences between males and females. Males tend to have thicker skull bones. This also holds true in this particular situation. So our orbits, okay. That's what we call our orbit, and the orbital shape we're looking for. So I'm going to go over to the right orbit here. And we'd kind of look at this and say, what type of shape are we seeing? If I was looking at this, I would say that that's type of an oval shape we see there with a little bit of a squared off edges. So oval squared off edges, descriptive language, that's going to help us understand what we're looking at, and the reader as well. Number six, this is our mastoid process. So the mastoid process right here, okay, and that's going to help us determine ancestry and sex of the individual. Number 7, okay, so the auditory meatus. Is it round or is it oval? And I pointed out that that would help us identify the ancestry of the individual in question. This is the zygomatic extension. So the zygomatic extension. So the zygomatic bone. So this is the zygomatic bone. And we have a suture. Actually, I'll use black so you can see it better. So this is the zygomatic bone here. And it ends right about there. And then we have the arch. We continue to call that the zygomatic arch. And we're interested in understanding or visualizing, see how far it extends past that auditory meatus. And that's going to help us identify whether or not the individual is male or female. Number 9, this is the nuchal. I need an H there. That nuchal crest, which is going to be important in determining whether or not the individual is male or female. And number 10, we have the Inca bone, whether or not it's present or it isn't present, right in that area, right around there, and that can help us with ancestry. Okay, so these are the landmarks that we find in that neural cranium. Let's move on and take a look at the visceral cranium. So I'm going to stop here for a second as well and just, show you that really part of the process of studying and understanding stuff is reviewing, and even if it's coming back to an image like this and going through it really quickly, right? For example, and just with letters. So this is the parietal bone, this is the frontal bone, this is the temporal bone, this is the occipital bone, this is the mastoid process. This is the zygomatic extension right here, the auditory meatus. What else can I point out? The mandible, the maxilla, the zygomatic bone, the nasal bone, and we can continue to work through that. So we're looking for up here frontal bossing. We're looking at the gallbella in this area. We're looking for a supraorbital brow ridge in this area. We're looking for a superciliary arch in this area. And if we continue down a little bit further, which I'm not going to because we're not there yet, we'd obviously continue with that. Here I can see the lacrimal bone. Here I can see the sphenoid bone. Here we have a nuchal crest. All of this is really good for studying and just kind of going through it really quickly, and then do the same for the front as well. Okay, so we're moving on to the visceral cranium, the landmarks, and I just want to point on before we get a little bit further down. I pointed out that the mandible was not part of the cranium, but I have included it on this chart just as a matter of ease. Okay, so our nasal bone, we are going to find that on our nasal bones. All right, so that's the location. It's our nose. And this is going to help us identify ancestry. The nasal aperture. So this is really an area that's a composite again. It's constructed as a result of the nasal bone, the maxilla, and it helps us identify the ancestry of the individual. A student famously referred to this as the nose hole. So, when you're cutting a pumpkin, for example, and everybody likes to put that triangle in for the nose, well, really, some noses do have that triangular shape. Some noses have more of a rounded shape. So that's the nasal aperture, and that's going to help us with ancestry. The nasal sill or guttering, this is something that we find on the maxilla, and it's going to help us with ancestry. The nasal spine, we find that on the maxilla, and that will help us with identifying ancestry as well. The malar tubercle, we're going to find that on the maxilla, and that's going to help us with ancestry. The mental eminence, we're going to find that on the mandible, which is not part of the cranium, it's part of the skull, and that's going to help us identify sex. And here I've got a couple other ones, and depending upon whether or not we have time, the palatine bone and the dental arcade, so the roof of your mouth, that's the palatine bone, and your dental arcade, the shape. Some populations, for example, they do exhibit dimorphism. There's morphological or physical differences. So some individuals, their teeth populations, they tend to be fairly square from front to back. Some individuals, they tend to curve inwards, populations I should say, and some individuals tend to curve outwards. So by looking at that, that can help us identify the ancestry. And with respect to the palatine bone, so the roof of your mouth, so if we're looking at the roof of the mouth here, our palatine bone, it's sutured, and depending upon how it's sutured here, we see a few different, I'm not going to draw them all in now, but just to give you an idea, we do see a few differences in patterns. That's going to help us identify ancestry as well. So that's a good area if it's available and you can see it to take a picture of and observe. And like I said, as we get further into the course, you're going to learn a little bit more about the significance of those particular landmarks. Okay, so referring to the table we just completed, we can go through this pretty quickly. Number one, we have the nasal bone. All right. Number two, we have our nasal aperture. Number three, we have a nasal spine, which is going to help us, as we pointed out, with ancestry. A lot of these have to do with ancestry. Number four, opposite side, this is where we're going to find whether or not we see nasal guttering. Kind of think of a bowling alley where it has a kind of groove versus a sill where you have a windowsill, a blockade, something that prevents stuff from kind of moving in there. Not that that's the function. Number five, move up to the top. This is where we see the malar tubercle. And number six, the mental eminence, which is going to help us really out of all of these. It's the only one that's going to help us identify, estimate the sex of the individual. Okay, so we've talked a little bit about teeth, and I've talked about a few things. So here we're seeing the palatine bone. We don't see in this particular image any suture, but here we would normally see some type of a suture going across there, and that could help us identify ancestry. And here we also see a particular pattern to those teeth where they're flaring outwards, that dental arcade, rather than being pretty straight in towards the back or the dorsal area of the body. And in fact, we use different terms. I won't go over, but you have what's referred to as lingual versus buccal. So some different terms when we talk about odontology or dentistry. I won't get into those. Sorry, I did. Okay, so going through our teeth, we've looked at these. So incisor, canine, premolar, molar, molar. I'll say PM for premolar. And we see that exact pattern represented in the second quadrant, the maxillary quadrant. Incisor, canine, premolar, molar, molar, and these are also note that they're adult teeth, and that's something that we will also use or can use to estimate age at death. Here, I want to briefly talk about evolution and culture as it relates to a few of the things that we've already discussed to our anatomy, and I've already pointed out this. When we look at hominoids, or we want to say ancestral hominoid is a species that's ancestral to humans. They may or may not be directly related to us. Um, a chimpanzee, it's a hominoid. It's related to us. We do share an evolutionary heritage. We did come from the same species at one point of time. That branched off to chimpanzees and then leading to us. In contrast, anything that fits within our own evolutionary line we call hominin. So that's a direct evolutionary relationship. Either way, when we see these homologies we've talked about, we see them in a variety of ways, and it allows us to say, yeah, we're related to chimpanzees. We're related to gorillas because we share homologies. Right? And that's shared ancestral species features. So the dental formula we share is 2, 1, 2, 3. That's the pattern. We also share a cusp pattern. So here on the surface of our teeth, so let's go to the top. We have some space there. We do see different patterns. We have what's referred to as a Y5 pattern, and we also see that within. So it has kind of a Y groove there, and 5 cusps, right? 5 different cusps. And that's something that we also see with among the other apes. It allows us to say that we are related. Here's a biocultural bit, for example, and we've talked about this as well. Adoption of farming, it led to smaller jaws. This ultimately led to a mutation where we started to see larger brains, and selection for wisdom teeth begins to disappear. Don't need the wisdom teeth. We did need, wisdom teeth, that third molar. In a large jaw, if we say what's the function, right? Large jaw, I'm just going to use increase air. Large jaw and extra molar. That's really for mastication. Or chewing, breaking down food. As food became softer, we just didn't need that. And as we were starting to incorporate more meat into our diets over time, our brains were getting larger, and it just made a lot of evolutionary sense. So wisdom teeth, the lack of, ironically, has to do with a bigger brain. Looking at our teeth, we can, and as I pointed out, we can figure out time since death. Okay, or age at death, how long an individual has been deceased based upon the age, right? So did they die at in their teens? Did they die in middle adulthood? On and on. The other thing that we can use teeth for is we can pull out something called ancient DNA. And ancient DNA can help us establish genetic relationships. Okay, and we can see clearly in a lot of situations what type of I don't want to get into too many terms, haplogroups, haplotypes, the genetics. So these are the types of things that, for example, Ancestry.ca traces, and National Geographic, they did it for a while, and they look at our ancient DNA and see what populations we originated from. What's there in our DNA, genetic markers. So we can use it for that purpose. It can also tell us about disease as well, because not only do we see, for example, human DNA in our teeth, which we would expect to be there. Okay, so definitely we're going to see human DNA. But if you're really sick and you had, say, a massive malarial infection, you're also going to see the DNA for the malarial protozoa, Plasmodium falciparum. So we can see the DNA there of a protozoa, a particular type of parasite. And it can allow us to say, well, this individual died as a result of malarial infection. Or if you've followed anything that's taken place at McMaster University, Hendrik Poinier down there, he also looked at teeth, and through DNA analysis, he was able to determine that in the 1300s, that big, huge plague that ran through Europe, it was in fact a plague of Yersinia pestis, bubonic plague, the Black Death, because he could see that DNA in people's teeth from the particular bacterium that was responsible for killing them. The other thing that we can use teeth for is we can see whether or not somebody was sick in a different way. We can look at the surface of the tooth for something called linear enamel hypoplasia. So this is in our textbook, and this is just a review. These are those lines that run across teeth that help us understand a little bit more about malnutrition. So it is something that emerges if an individual is malnourished. Our teeth, and while I've pointed out that they can help us looking at the DNA of the teeth, ancestral connections, dental morphology, or the shape, write that in there, the shape of our teeth and the shape of our K, they do differ by ancestry. And here, for example, when we're looking at the morphology, we're looking for extra cusps, for example. So a cusp, if you feel the surface of your tooth, okay, on the crown, those little bumps that stick out, that's called a cusp. If you take your tongue, and you run it around, I'm doing that now, so that's why I sound a little funny, run it around your premolars, if you feel something sticking out of the wall, an extra cusp out of the wall of one of your premolars, usually the first premolar, it's called cusp of Carabelli. We find it in Hungarian populations, and it actually traces its heritage to Mongolian migrations that moved through Hungary during the time of the Mongolian invasions. Okay, so very important teeth. The other thing that we can use teeth for is we can achieve, it's probably the best way to achieve positive identity. A little black here, especially if the individual has a filling, because a filling is just as good as a fingerprint. Right? It's just as good as that fingerprint. So that filling, they are unique. So there's a number of things that we want to look at when we're looking specifically at the teeth. It's called forensic, right up into the top right-hand side. Forensic odontology. But you knew that because that's in your textbook. Okay, so moving on to the body, away from the skull being part of the axial skeleton. Here we do have part of the axial skeleton and also the appendicular skeleton. The major bones that we're going to be concerned with that I need you to remember, include the clavicle, the vertebral column, the ribs, and I'm not going to make you identify every rib, the first rib, the last rib, the floating ribs, just that we have ribs. The sternum, that's going to be more important. Humerus, radius, ulna, innominate bones, the sacrum, the femur, the tibia, fibula, where the carpals, tarsals, phalanges, and metatarsals and metacarpals are. As for function, again, this is pretty obvious: movement and support, protection, and also storage of really important stuff in our bones, for example, minerals. Okay, so now we are looking at what's referred to as the shoulder girdle. So it's how all of these bones, and we'll go over them in a second, articulate with each other. If you're looking at it, you should be thinking already, what view are we looking at? We are looking at anterior or ventral. And is it a right or left assembly? So if you're thinking right, you are correct. Okay, so we're looking at a right assembly. Okay, so the bones that make up the shoulder girdle include the clavicle, the scapula, and the humerus. Nothing funny about that. So our clavicle, so one of the reasons we need to do this is because sometimes we do need to identify whether or not we're looking at right or left bones, and when we find them in isolation, we need to be able to articulate and say, okay, we're looking at, for example, the sternal end or the sternal facet, and the sternal facet is something that is medial. Okay, so the sternal facet here, that's towards, if in anatomical position, that will articulate with the sternum. So we don't see the sternum. So this particular area right here, I will use some blue. That's the sternal facet. If we begin to move, and we do move laterally away from the medial aspect or the sagittal section of the body, and we start to move laterally, we get to the area that the clavicle articulates with the scapula. And so you can feel this on your own body. If you feel your shoulder right now, that area that's sticking out, that is the acromion process. Okay, that's right here. And that is where the sternum and the scapula articulate. So the name that we have there is the acromial facet. And underneath the sternum, the clavicle, right under on the underside or inferior, it articulates with the coracoid process of the scapula, and we're going to find the conoid tubercle right underneath of there, and that does help us identify whether or not we're looking at it right or left, because it allows us to say if we see the conoid tubercle, we're looking at it inferior, then we know, for example, that it's belongs to the right side of the body. Other elements we're seeing here, so the humerus, we're seeing the proximal humerus and the head of the humerus, the surgical neck of the humerus, the greater tubercle. We'll go over that in a minute, and it articulates with the glenoid fossa. And here we have a ball joint. Really, that's what it creates, is a ball joint. Okay, so let's move on and take a further look at the bones and the landmarks that are going to help us gain an understanding of the skeleton and reconstruct life from it. Okay, and remember, you have materials that you can work along with me. So here we have the clavicle. And I pointed out that we have two clavicles. And one of the objectives here is we're looking at it, we need to identify whether or not we're looking at a right or a left. Are we looking at this from an inferior or a superior view? Okay, so our top image, this is a superior. This is a superior view, so we're viewing the top. And on the bottom, this is inferior. Okay, both clavicles. And if this is superior, this is the sternal facet. And number 2 would be the acromial facet. And now looking on the underside, we have the conoid tubercle. And this tells me, and one of the ways that you can think about this, you can feel your clavicle, and you can see how the shape of it. That's one of the ways. One of the methods that I used when I was a student, I looked for the flat side. Okay, so the side that didn't have the conoid tubercle, I knew that that was obviously the superior or the top end of it, and I pretended that this was a handle of the hockey stick, and this was the acromial end was the stick part where we'd hit the puck. And I would hold it and I would say, okay, so now I'm going to hit the puck and I have to shoot left-handed with this one. So moved it as I would if I was hitting a hockey puck, and kind of moving it into place, and it moves up to the right-hand side. So we are seeing it as it would be from a top view. And here, as pointed out, we're looking at the right side of the clavicle. So here are the image that you have. So right here, this is number 3. This is the conoid tubercle. So this is a clavicle. You know that immediately. I'm looking at 2 different views here. So I'm going to say that this is a superior and inferior. The conoid tubercle, so if that's the conoid tubercle side, this is the acromial facet, and therefore this over here needs to be the sternal facet. Okay. And that's exactly what we're looking at. If we know how the movement of this is the, again, this is the sternal facet. So this is our superior view, and this is the acromial facet that would articulate with the acromion process. Therefore, we are looking at what side? If this is the sternum over here, if we put that into place, we're looking at the right-hand side of the body. Okay, so we were looking at the clavicles. I'm going to highlight them in green on the left-hand side of our screen. There they are, and they articulate with, right up at the top. Okay, the sternum. So we're looking at a mostly articulated sternum. The manubrium, okay, it hasn't fully articulated with the sternal body. Okay, so those are the 2 terms we need. And down at the bottom, we don't see it present, and quite often this breaks off in archaeological or bioarchaeological finds, recoveries. This is the xiphoid process. And up at the top, the area that articulates with the clavicle is called the jugular notch. Okay, let's move on to the sheet we're working along with. Okay, so at the top, as pointed out, we have a jugular notch. We have the manubrium. We have the body. And here we have the xiphoid process. And our bone, therefore, is the sternum. There's only one side, so it is what it is. We don't need to fill that out. And the view, we're looking at the front of this. So this is anterior or ventral. I'm also going to point out something else here if you haven't noticed it. Right there. That's called the sternal foramen. It's a little hole, and it can be used in some situations when it is present to identify the sex of the individual. Okay, so here we have the scapula. You can work along on your sheet, and then I'll come to that one and we'll see if you got it right. The scapula, it articulates with, and we've pointed this out. If I go over here, we see the clavicle, and we've pointed out that the clavicles, they articulate with the sternum. So this is how all of these pieces fit together. Let's go through the main parts of this. So what are we looking at here? Scapula, obviously. In the first view that I'm seeing, I'm just going to use ventral now, because you know that that's anterior. And the second view that I'm seeing here, because I do see a spine, I'm just going to put a red dot on that spine. That's dorsal. Okay, so let's work from number 1. This is the acromion process. Number 2, we call that the glenoid cavity, fossa. Number 3 is called the coracoid process. And number 4, we have to go to the dorsal side to see that. That is called the spine. And number 5, that's referred to as the body of the scapula. Okay, let's move over to the drawing, and I'll go through that, and we'll correct it if necessary. Okay, so your view is a little bit different than mine, so we should pretty much have the same stuff. S-C-A-P-U-L, the scapula. The view. Okay, this is where it was different. So I had both views. I had ventral and dorsal. You have dorsal. Okay, so you got that great stuff. Therefore, if this is dorsal, we're looking at the back. This is exactly the way it would look if it was on my body, as I'm pointed to this. So this is the back. This is the right scapula. And as for our various aspects, sorry, my pen stopped working. The acromial process, the glenoid cavity, we've got a spine, we've got a body, and we have the, I'm going to try and squeeze it in here, the coracoid process. Okay, important to have your notes. Make sure you have those notes. You're going to need them, right? It's open book test. Okay, moving distally down the body in anatomical position, we come to the elbow joint, or what we call the humeral ulnar joint. And it's made up of the humerus, the ulna, and the radius. Okay, so 3 bones here that we need to be aware of. So if this is the humerus, we're looking at the distal humerus. In this area, we are looking at the proximal, because this is the top, so the proximal ulna. And moving over to the radius, we're looking at the proximal radius, because it's towards the top of the bone. Okay, so a few landmarks here then. We have the medial epicondyle as such, the lateral epicondyle. So this is towards the outside of the body, and the medial, this is towards the inside of the body. If we go up further, the humerus would be articulating with the scapula on this side in that glenoid cavity. Okay. Other landmarks we need to be aware of. So the trochlea, right there. The capitulum, and the capitulum articulates with the head of the radius, and the ulna articulates with the trochlea, and the area here is called the trochlear notch. Okay, let's move on and take a look at these bones individually. Okay, so here we have the humerus. I guess that's why we call it the funny bone. Okay, the humerus. So starting off at the top, we have an area. I'm just going to try to squeeze it in here. Maybe I'll go to the left. This is what we refer to as the head of the humerus. And this is the greater tubercle. This is the shaft, number 3. Moving down to number 4, we have the lateral epicondyle. Five, we have the capitulum. 6, the trochlea. And 7, we have the we're on the medial epicondyle. And number 8, this is the ulnar notch. Okay, now let's take a look at your slide and see what you came up with as you were working along, and we'll fill that out together. Okay, so we have the head and the greater tubercle, the shaft. As we move further down, we have the lateral epicondyle and the medial. The medial epicondyle. So the medial epicondyle is on the same side as the head, right? So that's one of the ways that you can remember, because the head is always going to be medial. So if this is the medial epicondyle, the lateral epicondyle, then, and let's go over to the larger image. So this is the lateral epicondyle, the medial then we have the capitulum, because the capitulum is lateral, and then the trochlea, which is medial. Okay, so capitulum lateral, trochlea medial. Looking at this, we're going to fill this in. Say the bone is the humerus. The side, this is a left. And our view would be ventral. Okay, let's move on to the next bone. Okay, so here we have the radius. Number 1 is the head. Number 2 is the neck. Number 3 is the radial tuberosity. Number 4 is the styloid process. And number 5, this is the ulnar notch. Now I have two of these highlighted in red because they do help us identify the side of this bone. So the rule is if we're looking at the radial tuberosity, so that's one of the things that we want to identify for figuring out what side of a bone. Are we looking at a right radius or are we looking at a left radius? I'm going to take a look at that radial tuberosity. And the rule is it's found on the side the bone comes from. So in anatomical position, we see the radial tuberosity here on the right side. Therefore, this is a right radius. As for the styloid process is found on the, I'll just say styloid process. We find the styloid process on the side the bone does not represent. So if it's found, for example, on the left hand side, it means that we're looking at a right bone or a right radius. And one of the ways that you can get at this is just hold your body in anatomical position. So the radius is found in anatomical position laterally on your body, and think that your thumb radiates out. So if I was to draw it in here, our thumb would eventually start to come out here, sticking out in anatomical position. That's where the styloid process is. So if it's found there on the left side laterally, that tells us that this is a right radius. Okay, so hopefully you got this right because your bone's a little bit different that you were working with, and let's go through it and make sure that we have it correct. So here we are looking at the radius. And if we're going to label it, which we want to do, we have the head, we have the neck, we have the radial tuberosity, and down here we have the styloid process. And we can't see the ulnar notch because it's on the back. So given that we can't see the ulnar notch, we know that we're looking at the front of this bone, or our view is ventral or anterior. It's up to you, whichever you prefer to use. Now, as for the side, so the side of this bone, remember the rule. I'm going to go over here. The rule of the tuberosity. So if the tuberosity is found on the left, that means it's a left radius. If it's found on the right, it equals a right radius. And the opposite applies with the rule of the styloid process. So if it's found on the left, it means it's a right. And if it's found on the right, it means it's a left. So here we find the radial tuberosity on the left side. We find the styloid process on the right side. Therefore, this is a left radius. Okay, so here we have the ulna. And the ulna, if we look at the body in anatomical position, we see that the ulna is more medial than the radius. The radius was lateral, so the radius is on the outside. And we can also see, remember that the thumb radiates out, so that places the radius where the thumb is, on the thumb side, in anatomical position. The ulna is medial. It's towards the inside of the body. Okay, so the basic landmarks here, we're going to have 4 of them, and 3 of them are going to help us identify the side. We start off with the olecranon process, and then we move to the trochlear notch, and then we have the radial notch, and the styloid process. Okay, so the radial notch and the styloid process, we're going to use those to help us identify what side bone we are looking at, a right or a left. And as I pointed out, that's really important to know whether or not we have two right bones, if we have two left bones, if we have three right bones. We start counting this to determine what the minimum number of individuals, the MNI, is as one of the first steps. And then we start to build up the biological profiles. So the radial notch here, the rule is, again, it tells you what side it's from based upon being opposite to it. So here, in other words, we see it on the left, right? So that means that this is a right ulna. If we see it on the right, that means it's a left ulna. And the reason why, if we think of what articulates here, this is where the head, I'm just going to kind of sketch it in here, where the head of the radius is going to articulate right in that notch there, right up here, a little bit low, right up there, that's where it's going to articulate. So therefore, if this is on the outside, remember it radiates, that means that this is a right ulna. Okay, so this is a right ulna we're looking at because that notch is on the left side. As for the styloid process, it tells us the side that the bone comes from based upon the side it is on. So the styloid process here, we see it on, we use that rule again, the similar rule rather than the reversed rule. So here we see it on the right-hand side, therefore it's a right bone. If we saw it on the left-hand side, it would be a left bone. Okay, so let's go and see what you've come up with on your chart. We'll make any corrections as necessary. Okay, we're going to do a little bit of forensic work here with this ulna, this drawing. And the reason why we go through this, so we've got the Alcarnon process, and then we have the trochlear notch. I'm going to go right down to the bottom, to the styloid process. And you might have shaded in, I'm going to go to a highlighter here. You might have sitting around here, you were thinking, was the radial notch. It can't be because the styloid process and the radial notch are not found on the same side. So the radial notch has to be over here. We can't see it. So when it comes to our view in this particular image, we don't have entirely a ventral. It's twisted a little bit, but we'll call it ventral anyhow. Mostly, we've had to do some work there. As for the side, okay, so the styloid process, the rule is it tells us the side that it comes from based upon the side it is. So here we see the styloid process on the left. That means that we're looking at a left styloid process. So we're going to go in and right left. And if we're looking at the radial notch, we find it on the right-hand side. And sometimes it helps really to label things on the side that they come from for those particular rules. So our radial notch, we see it on the right. That means that this is a left bone. The reason is, okay, so I'm going to go black and I'm going to just draw in a radius. So if I had a radius, it would be articulating right in this area. It would be the head, right? And it would come down as such. We get our styloid process. And what we know about these bones and the way that they articulate and the way that they sit in our body is the radius, it's found laterally. And the ulna is found more medial. Therefore, if this is radiating out, this is our thumb side on our hand. Our thumb is sticking out. Therefore, this is a left It's confirmed. So our radial notch, we can't entirely see it, but that's okay. And this is why we need to understand bones, because quite often, and this is really, it's a beautiful example. It caught me by surprise, but it's a beautiful example because in forensic work, quite often we don't see all of these processes and notches and whatnot. We have to know what would be there. Okay, so here we're looking at the hand, and I'm not going to test you on this. You know, there's a lot of bones, and the way I think about this is when we progress into university, if we're pursuing forensic anthropology, biology, and health studies, then we need to have a further understanding of it. But for our course, we don't really. And the other thing I'll point out when we're looking at this hand is not in anatomical position. So we're looking at the back of the hand. So this is dorsal, right, or posterior. The side, therefore, is right. And as for bones, I'm just going to write up here. You can write the hand assembly, but I'm just going to point out what the structure is ultimately, and not all of the individual carpals and the names for them, for example. So these are our carpals. Our long bones, so our carpals, and I'll just use this for a second to go over what we've talked about already. Our carpals, they're short. They're short bones. Our metacarpals, they're long bones. Okay, and when we get down to the end here, we have our phalanges. And phalanges are fingertips, right all there. They're also a type of long bone as well. Okay, so let's move on to rest of the body. There's still pretty much half of it to go. Okay, so this is what we refer to as our pelvic girdle. Okay, so that's the proper name for the assembly. And it's made up of, what are we looking at here? 3 different bones. So the sacrum, the innominate bone, I've got that written in, but I'm going to write it in again, because I've started something and I'm going to finish it. And the humerus, sorry, not humerus. Get rid of that H. The femur, okay. And a few joints. So the sacrum, it articulates with the innominate. We have 2 innominate bones. We don't see the other here. And we have a joint that's referred to as the sacroiliac joint in this area. The acetabulum, okay, so that's the socket. Okay, so the ball and socket joint that we find in our shoulder girdle. We also find that in our pelvic girdle. So this is the ball and socket joint. So the acetabulum is the socket. And we have the femoral head that articulates with the acetabulum. And this is usually the area that people have issues with as they begin to age. So when we talk about hip replacements, quite often it's a replacement of that femoral head and also the surface of the acetabulum. So in that type of surgery, just out of interest, they usually take off the head here, and they put in a new head made out of titanium quite often, and then a new surface in here. That's off topic, but just if you're interested in kind of what goes on, because we do see a lot of those operations. Okay, so we have our femoral head, and then we have something called the greater trochanter and our pubic symphysis. Okay, so this is where we do have that joint with that other, if I was to draw it in here, or the other innominate bone that makes up the rest of our pelvic girdle. And we're looking at the Not a distal femur. What have I written there? That's not correct. I'm going to change that right now. I don't know if I can. I can't erase it, can I? No. Okay, so I'm going to erase that. That's not a distal femur. That's the proximal end. That's proximal because it's closer to the center of the body, to the head, than the other end. So the distal femur we're going to find down that way. Okay. Okay, so here we have an innominate. An innominate bone, sometimes referred to as the os coxa. We don't call it that. Some other people do. Okay, so the other thing to remember here about the innominate bone, this is something you really need to focus on when you're collecting data, because it just provides that much information. So it's going to get you from, about 95 to 100% accuracy, even 96, using something that we call the Finis method accuracy when estimating sex. Okay, so this is something you need to focus on. I'm going to come back to the areas that you really need to make sure that you have good images of. Okay, so number one, this is the ilium. Just for orientation, this is the acetabulum. We've looked at that on the other page. Okay, number two, this is the pubis. Number three, we have the ischium. Number four, we have a couple of things here. So this is the area of the pubic symphysis. And we see what the pubic symphysis looks like medially from the inside, right over here. You can see that. We also have something called the ventral arc. In that area. Number five, we have the subpubic concavity. And I'm just going to add something here. This is called the ischiopubic ramus. Okay. And taking a look at that area is also important for being able to determine or estimate the sex of the individual. Moving up to number six, this is called the greater sciatic notch. Okay, so things that we can use here. I'm going to go to, let me get a highlighter and tell you what you need to focus on. So when you're estimating sex of an individual, one of the things that we want to take a look at, I don't like that color, it's not bright enough. Get rid of it. I'll go to yellow. Okay. You want to take a look at or have good images of the pubis, okay? This region in here, in the shape of the pubis, as you will learn, is going to tell you or give you information into the sex of the individual. The other area that I need you to take good images of and focus on, number four, okay? And this area that we're focusing on is the ventral arc, and in particular, I'm going to go back to black, it's right in this region. So you're focusing on a very small region, getting very good images in areas number 2 and number 4. So that pubis, the pubic symphysis, the ischiopubic ramus, you want to get good images of. Moving down a little bit more, we want to get good images of that subpubic concavity. And really what we're seeing here is you're kind of seeing a little bit of a concavity It's exactly what it is. It's a groove upwards, okay? The other area that I need you to focus on for taking pictures, data collection, go back to yellow, is going to be that greater sciatic notch. It's going to tell us a whole lot about the sex of the individual in question, okay? So things that we need to focus on, again, I'm just going to come back to it with some white here. So like I said, I want you to make sure that you're taking images of the pubis, the area around the pubic symphysis, the subpubic concavity, greater sciatic notch, and in around this region of the ischial-pubic ramus. And you don't really need to go much further than that. That provides us with all of the information that we need to get at a very highly estimate for the sex of the individual. Okay, so your image here, we have an innominate bone, and the side, we'll come to that. As for the view, the first one, this is ventral, and the second one is it's a lateral, dorsal. So looking more towards the back, the buttocks in this region here. We're going to find the buttocks. Okay, so we have the ilium. We have I pointed out the acetabulum. I pointed out the pubis, and I pointed out the pubic symphysis, or connection. We don't use the word connection, but that's really what it is. The ventral arc, the subpubic concavity, and this area, hopefully I can write on this in red that it'll work. Issue, no, that's not going to work. I'm not even going to try. I'll have to draw a line. So the ischial pubic ramus, kind of right in there, that section of bone. And what else did we have here? Oh, I wanted you to be able to identify the greater sciatic notch. And as for the side, looking at this, well, this has to be a right side because the acetabulum, it's on the left. Okay, so that means that if you put yourself into that anatomical position, we're looking at the right hand side of the body, because that's where the femur is going to articulate. So here we have a right bone in both instances. Okay, this is the sacrum. I don't have a whole lot to say about the sacrum. We have the body and what's referred to as the coccyx, or the tail bone. The coccyx can be important in helping determine or estimate the sex of the individual. So I would take a picture or note of whether or not that coccyx, if it's sticking inwards or if it's pushing outwards. And it's something you can even feel anatomically. Yeah, you can, but I wouldn't recommend that. Either way, you can feel the coccyx. It tends to stick outwards on female, so it has a more of a dorsal movement, whereas men it projects inwards, um, ventrally towards the inside of the body. Okay, so here the sacrum. The side, there's only one side. Our view here is ventral, and I noted all that we really need to hear is the body of the sacrum and the coccyx, or what's referred to as the tailbone. Okay, so here we have the femur, and okay, our main points here. Number 1, we have a head. Number 2 is a neck. Number 3 is referred to as the greater. The greater trochanter. And moving down, we have a lateral condyle. And we have a medial condyle. Yeah, so looking at this particular bone, we're seeing both views. On the left-hand side, we see ventral. On the right-hand side, we see dorsal. Oh, number 4, totally missed it. We have the lesser. The lesser trochanter. As for using this in this bone, any particular bone on its own, you got about a 50-50 chance of being accurate when it comes to the sex of an individual. But the thing to keep in mind is length. What's the length of that bone? So, when collecting data, there's probably going to be some tools there, and I would take a measurement of the bone and then see where that falls. Are we looking at somebody that is within the male range length, because our femur being the longest bone in the body, it's going to tell us a whole lot about stature, and generally speaking, men tend to be taller than female, so it does help give credibility to that estimate that we have. So that's one of the things that I would want to be interested in, the length of that bone. Okay, so here is your image, and let me just orient myself here. Yeah, okay. So our bone is a femur. We have a head, and we have a neck, the greater trochanter, a medial, and a lateral condyle. If you're looking at this saying, oh, I've got my condyle on the opposite side. Well, no, not really. It might have been because you weren't paying attention to some of the details. The medial condyle is always on this side, and I pointed this out earlier as the head, because the head is a medial structure, right? So the medial condyle has to be on the side of that head. So looking at this particular bone, this is ventral, because we can see that greater trochanter, and our side, this is therefore going to be left. Because of the way it's going to fit into the acetabulum. So this has to fit into the acetabulum. So if you just kind of imagine putting that picture up against your body, that is a left femur. And the other thing is that can help you out with the siding of it, is on the back of a femur. I'm going to try and draw it here, where we have the condyles, and say that we have let's say we have our medial condyle this side, and we have our lateral condyle on that side, which end up seeing as this large kind of fossa, where you do get your muscular attachment on the inside. So there's a large cavity there. We're not seeing a large cavity there. What we're seeing is, I'm just going to add one more thing here. We're seeing a patellar surface or facet. Okay, so that's where the kneecap or the patella is going to articulate. Okay, so here we are with the femoral tibial joint. So we're moving further down the body distally, and the components we have of our femoral tibial joint include the femur, our tibia, we have the patella, and the fibula. Okay, so those are our main bones: the femur, the tibia, the patella, and the fibula. As for some of our articulation points, or our landmarks, I should say, so we have our distal femur here, okay, and we have our medial condyle. So here's the medial condyle, and on the outside we have the lateral condyle. So being the medial condyle, we can imagine that if we were progressing up the shaft of that femur, we're going to find the head on this side, because that's also medial. Okay, so but that's going to be further up. So we have our medial condyle, we have the lateral condyle, and similar structures on the tibia. We have the medial condyle on the proximal tibia, so our medial condyle. And on the proximal fibula, we have our head. Okay, so we're going to go through these bones, and that will pretty much wrap it up for our human osteology, and you'll have a strong appreciation for the areas that you do need to focus on when collecting data. Okay, so here we have the tibia. And the tibia, the main components that we're going to need to know for this, going to be the medial condyle. So their medial condyle, and the lateral condyle. And right over here, medially, we have the medial malleus, or hammer. That's your ankle. That's your inside ankle. That's another way of thinking about it. So looking at this, if this is medial, the medial condyle, and that's our inside ankle. I'm going to go over here, and I'm just going to highlight it. If that's the inside ankle, number 3, then that's telling us that this is a right tibia in ventral perspective. Okay, so let's take a look at the diagram that you have as part of your resources. Okay, so this is the tibia. We should have that. And one of the easy ways to recognize a tibia, I'll erase this in a second, is it does have that T shape. I can leave that there, that's fine. That has that type of T-shaped structure. One of the ways I just remembered things when I was younger and in school, stuff like this helps. Let's first identify the medial side. And the easy way to do this really with the tibia is look for the medial malleus. And that's it right there, our ankle. So if that's the medial malleus, then this needs to be the medial condyle. And if this is the medial condyle, then this needs to be the lateral condyle. Our view, it's ventral. Okay, one of the things that we are seeing here, maybe I do need to remove that. One of the things that we are seeing that helps us identify whether or not we're looking at the front or the back, ventral or dorsal, is going to be the tibial tuberosity. And you can feel this just underneath of your knee. So if you find your kneecap and just go down a little bit, you can feel that tibial tuberosity there. So we're ventral. And if this is medial, over here, this is our medial malleus. This needs to be a left tibia. Okay, because this is the inside, the ankle, and our fibula is going to be on the other side. On the outside, it's going to be lateral. So this is a left. One of the ways that we can define it is based upon the medial malleus. The side it's found on identifies the side that it comes from. So we see it on the left, therefore this is a left tibia. Okay, so we have our fibula, and we see the location of the fibula, and they are found laterally, whereas the tibia, they're medial. Okay, the tibia is there. Laterally, we see the fibula, and the fibula forms the lateral or the outside portion of our ankle. So I'm just going to go down to number 2. I'm going to start here, in fact, because that's lateral. So now we're going to call that a lateral malleus. Number 1, this is the head. If this is the lateral malleus, this is the outside ankle. This needs to be the one that we're looking at in ventral perspective. This is going to be right. Okay, so let's move over to your diagram and let's see if there are any differences and any corrections that we may need to make on yours. Okay, so we should have fibula. I've written in the head and the lateral malleus as such, the head as such. So this is on the right side. Therefore, this is a right bone in ventral or anterior perspective. That's a pretty simple one. The last thing we're going to wrap up with refers to our tarsals, our metatarsals, and the phalanges, our feet. Okay, so our feet and the main bones, and again, the same thing applies with respect to our hands, because when we move to these types of short bones that we're going to find in here, our tarsals, it's pretty complicated, I think, for a college course, especially a general education elective. We don't need to be able to identify all of our tarsals, but we need to know that our tarsals are found in our foot. And then we have our metatarsals. They are long bones. Our tarsals, we pointed out in the same way that we talked about our carpals, they're short. And then we have the phalanges. Same as the fingers. are tips, and they're a type of long bone as well. So here we are looking at, if you can figure this out, what foot are we looking at, right or left? This is a right foot. And our view is going to be lateral. You're wondering why? Well, this is the pinky here. So we're looking at a side profile of our foot. This is the big toe over there. So yeah, this is our foot. And as pointed out, the bones that we need to remember, we're just going to be concerned with those categories: the tarsals, the metatarsals, and the phalanges.